That's great. Well, it is 2:30 and we'll get started because we have a lot to discuss today. Thank you and welcome to everyone who has logged on to share this next hour for our 21st presentation of the COVID Healthcare Coalition response series. Today we'll be discussing COVID-19 testing and reinfection. And we have a great lineup to discuss that. But first I'll send the mic over to John Halamka and bring us up to date on what you're seeing around the country, John.
Well, thanks so much. And I begin each one of these webinars by describing the 5 phases of COVID and how we've gone from isolation to testing and contact tracing to pre-vaccine return to work, post-vaccine return to work ahead, and the new normal. As we go through that continuum every week, we're progressing farther and farther through those 5 stages. And certainly, as you look at the last week, a lot of activity on convalescent plasma, hyperimmune globulin, still recognizing that that is our best hope for treatment before we get to a clinical trial proven vaccine. And CFX, I use the term clinical trial proven vaccine. One cannot declare victory because you have a not clinical trial proven vaccine in Russia. And so Operation Warp Speed has taken a great interest in this. This involves Dr. Fauci and Dr. Birx, Steve Hahn, Francis Collins, the Surgeon General. You've seen a large number of public service ads show up on billboards and television, encouraging individuals to donate and building the convalescent plasma pool. To give you some statistics, we've had 225,000 prospective donors go to the Fight Is In Us website thus far to go through the bot to donate plasma. So the social media campaigns and the public service announcements are certainly generating interest. So we'll see, I hope, maybe this week, the EUA issued on the use of convalescent plasma, in which case then certainly more and more demand will happen, as well as we're seeing clinical trials now try to get us more evidence about its efficacy and supporting the hyperimmune globulin development process, which will also go into clinical trials shortly. Much of the discussion this week has also been on back to school and back to work and all the elements involved. And what's so important about this coalition is the situational awareness that we are publishing on the c19hcc.org website to show you where we are at each county level. And where I must encourage all of you to read the MITRE authored report on back to school strategies for the nation, all based on evidence, all based on science. And you'll see in Appendix A that a decision maker at a school district can look at, is that district red, yellow, or green? What should they open? What should they not? So really a combination of data on our website as well as advice from experts is going to make back to work and back to school much easier. But remember, testing will be an incredibly important part of that process, and hence the reason we have the esteemed panel gathered today.
That's right. Thank you, John. As we are caring for patients and particularly healthcare workers, we're constantly asking the question, when can this person potentially return to their work site? And the opportunity now to use a combination of testing is before us. The science is still emerging. And so we're going to start today's conversation with a presentation from Dr. Bill Morice, president of the Mayo Labs. And then we'll have some insight from Dr. Ryan Lugambiel and the electronic health record data team that's been putting together a large data set to help elucidate this area. And his colleague, Melissa from EPIC, will be helping to present that information. And then our final speaker will be AJ Venkatakrishnan from Nference, that has also done a tremendous and recently published analysis about PCR testing and its synergy with serology testing. So Bill, why don't you go ahead and take it away?
Okay, thanks a lot, FX. So I just wanted to give people sort of an overview of diagnostic testing for COVID-19. There's still, although this has been in the press and in the public conscious for the last 6 months, still a lot of confusion, and maybe it's because of those things that there's still a lot of confusion about the testing and its role in the management of the pandemic and what it can and can't be used for, and its strengths and weaknesses. I think one of the things that really, as a fundamental, is to remember that the performance of any diagnostic test really depends on what it was designed for and what's the nature and the prevalence of what you're looking for in the population being tested. And so, going back to the basic 4x4 sort of box that we put together to understand what is the positive and negative predictive value of a test, and that this is truly predicated on the prevalence of the disease. What we have seen on the molecular side, there are some concerns that has to do with the time course of the disease and when you test, and we'll go into that in just a second. I think where this has really come into play is in serologic testing. So where we saw serologic tests that seem to have relatively high performance from an analytical perspective, being 95% sensitive and specific. If you start applying those tests in areas where there had not been much infection, you actually had a much higher chance of a false positive than a true positive, and some of those things that started to undermine people's confidence just in diagnostic testing in general. And so that's one of the things that I'll always go back to when you start thinking about the testing. When you think about the tests, the things that we have in our armamentarium now are pretty similar to, honestly, what we had 4 or 5 months ago. There is the PCR test for the virus itself, right? The test for the SARS-CoV-2 RNA in a clinical specimen. The gold standard for that specimen is still the nasopharyngeal swab, because it has the highest probability of actually having virus in it to be detected. There are specimens that are more convenient, such as a spit test, a saliva-based test. So far, those alternate specimen sources tend to have less viral load and therefore a little bit more prone to false negative results. By and large, the challenge with the molecular testing for the virus revolves around false negativity, and there's a couple of reasons for that, including when the patient gets tested relative to when they're infected, which we'll go into in a moment. The next class of tests really are the serologic tests, or the blood tests to measure for antibodies generated in response to the SARS-CoV-2 infection. There's really two main forms. One is the ELISA, the in-lab test, which is done at high throughput. There are some tests that are isotype specific for IgG and IgM. There are others that are IgG only. There are some that are sandwich reagents, such that they test for both but don't distinguish. All are predicated on detecting the creation of antibodies to the SARS-CoV-2 virus. Again, this was one that early on, some of the reagents introduced into the clinical labs had good but not great sensitivity and specificity, and the application of those tests in areas where there had not been much infection led to some false positives and some undermining of the confidence in the tests. The other tests which were on the market and still are on the market are the so-called lateral flow tests, or rapid antibody tests, finger prick, self-read, with it's like a home pregnancy test. Those can be high quality and can have a similar performance to the in-lab testing. Again, there was a flood in the early days on the market, both here and internationally, on tests that had not great analytic performance. We did some evaluation here. We had one test in particular that showed a positive result with water. So there were some pretty specious tests out there in the lateral flow world. That has been addressed, and to a large extent, and I do think that these will have a role going forward, because really the need to get as much data, to John's point earlier, as possible around the virus itself and its spread. The other kind of class of tests to just discuss quickly are antigen tests. Antigen tests are for the virus itself as well, but they are for the protein, not the nucleic acid. These typically are not of great use in respiratory viral infections, just because they tend to have pretty low sensitivity, lower than the PCR. They are much more amenable, though, to point of care diagnostics, and so there's a lot of work happening in these. There are two on the market right now, one from Becton and one from Quidel. So far, there's still relatively limited application, but a lot of interest in a point of care antigen test that could be self-read like the serology test. And then the same goes for PCR or other molecular methods like LAMP, which are non-amplifying. The challenge with all of these tests to detect the virus, and part of why they get something of a bad name, is it really depends on when you test the individual after they have been exposed. So what in this graph really shows nicely the chance of a probable positive test by PCR using a nasopharyngeal swab after infection. And you can see here in the very early days after exposure, in the first 4 days, you're actually more likely to have a negative test in someone that is going to eventually be positive than you are a positive one. That is so important here because it's really in that window that we want to start to detect people when they're pre-symptomatic, because it so happens for this virus that this day 5 window, when they still might test negative, is actually when they're most apt to be shedding the virus and spreading the virus most readily, right, or most effectively, if you want to look at it through that lens. So again, understanding where we test, the viral RNA tends to disappear after seroconversion. That's shown here. So if this is day 0 for viral infection, and this blue line is basically the ability to detect the virus itself, what you see anywhere from about 5, as early as 4, but typically by around day 7, you start to detect seroconversion or the presence of antibodies in the blood. Typically, those will be, the first isotype will be IgM, and then we'll convert to IgG somewhere around day 10 to 14. There are definitely deviations from this, though. There are some patients that never get an IgM. They go straight to an IgG, or they get IgM and IgG synchronously as opposed to asynchronously, as shown here. And also, there are some, particularly those who are minimally symptomatic or asymptomatic, that might not have much of a seroconversion at all. So the other thing to take away from this, which is relevant to reinfection, is that it tends to be about anywhere from 10 to 20 days after the appearance of antibodies in the peripheral blood that the virus becomes undetectable in the nasopharyngeal swabs. And again, as you'll hear from AJ, this is represents the average experience, but certainly is not universal. So clearly there's a need for innovation, and I think innovation in diagnostics will answer a lot of the questions that are still open around the whole concept of reinfection, as we're going to delve into with our other speakers. Clearly, right now what we have by PCR is a qualitative test, for the presence or absence of the viral RNA. Understanding the actual quantity of detected RNA is probably going to be really important. We do have introduced a digital droplet PCR, quantitative PCR assay. I think there are others that are working on this as well. This is going to be really important as we start to think about, the questions around what does a persistent positive qualitative test mean, which does occur, as you'll see. A high throughput antigen test, which may actually be more indicative of who's actually spreading the virus, is another thing that needs to be worked on. If you're really going to use it to detect viral shedding, as opposed to just viral infection, again, you'd want a sensitivity similar to the gold standard PCR. Again, the rapid diagnostics are going to be really important. Neutralizing antibody assays, of which there are a few now which are clinically available, are also important. So this is a variation of the serologic test in which you actually take the antibodies from the blood and show that it can block viral infection in vitro in some way. As it turns out, so this is really measuring for the bioactivity of the antibodies as opposed to their presence or absence. Again, really important for some of the convalescent plasma work, probably important for vaccine trials, and there might be actually some importance as well in terms of better correlating with who's actually not shedding the virus or not actively infected by infective virions because of the presence of neutralizing antibodies. There was one study that showed that neutralizing antibodies, even if they had a detectable PCR or SARS-CoV-2, they did not have infective virus that could be detected. So, and then last but not least, then, as we look at focus right now is on humoral immunity and measuring humoral immunity. We know that T and NK cells, innate and acquired immunity, are really important in essentially almost all viral infections. We just don't have a lot of great readily scalable lab assays for those. But I think that's going to be another area of innovation we're working on, and a lot of people are. Some of the early studies results from the vaccine trials suggest that memory T cells are going to be very important to measure. So that's something that's really actively being developed. It's just much more difficult to analyze than is serologic testing for humoral immunity. So I will stop there. Hopefully stop sharing my screen effectively. There we go. And give you back the virtual floor, FX.
Good. Thank you, Bill. Well, it is a complicated story, but it's emerging, and it is following the same pattern that other infections obviously have in the past with a antigen followed by an IgM followed by an IgG. And so how do we harness that into clinical decision making is going to be the challenge, and how do we get testing that has widespread availability. Usually with most other infections, this development period is over a much longer period of time, and the opportunity to refine these methods and these protocols are things that go on inside the medical literature and medical conferences, but now it's all out to view. Yeah. And it's been an interesting public discourse.
For sure, as I've said before, sorry, I didn't mean to interrupt this. We do this all the time. It's just usually not a spectator sport, number one. And then number two is that this virus is challenging, because where we want the diagnostics is in the asymptomatics, where we know it's going to be most challenging to actually detect the virus.
Right, right. Okay, that's great. And please continue to put your questions in the chat window, because we will have a good amount of time to discuss these. And I know there's a lot of interest in the nuances of testing and testing strategies. Ryan Lugambiel is going to speak next. He's going to introduce a tremendous body of work that's been going on for the last 4 months related to the collaboration of electronic health record groups, companies, and medical organizations to bring data to bear for elucidating the pandemic. So, Ryan?
Thanks, FX. Let me share my screen. So I am Ryan Luginbuhl. I'm a physician and principal for the Department of Open Health Services at MITRE. I co-lead the Coalition's decision support for social policy North Star, along with Monique Mansoura, and this includes the non-pharmaceutical intervention, waste-based epidemiology, analytics, modeling and simulation, misinformation, contact tracing, testing, and characterizing COVID working groups. And I also lead the optimization of clinical therapeutics and protocols working group and our characterizing COVID working group. So where do we start? So this all starts basically on April 4th, when we started with the convening of the nation's healthcare analytics platforms and EHRs to fight COVID-19. And we started doing federated real-world data studies. And so the first study we put out was inpatient famotidine. But fast forward, we started getting very interested in other use cases, especially as cases surged. We started to think about other ways that we could use the data, such as could we understand reinfection, which is becoming a more and more key question. So could we leverage the data to say, can COVID-19 reinfection occur after an initial infection? And so to do that, we started looking at a first and a last RT-PCR. No matter how many positives there were, we really want to see the first and the last, and then capture the days in between, plot that out. And so we gathered together and we started capturing those differences amongst the nation's leading EHRs and healthcare analytics companies. And we captured this curve, this is updated through the end of July with 56,871 cases of a first and last RT-PCR. And you can see the distribution in buckets of 7 days and the percent of the patients with those multiple positive tests. And so we're going to capture that distribution monthly going forward and really look at this tail here to see how it changes over time. And we think this gives us a sense, this separation between the two dates, of how long people can shed the virus. And then number two, whether reinfection is possible. There are 3 potential drivers for the distribution here. One, the distribution could be the length of disease in COVID-19, going from days to months. Number two, it could be due to a small amount here, especially the tail of false positives, which we'll look to in future months to see if it gets surpassed. And it could be reinfection. And we really can't quantify exactly which component it is. So going forward, we will update this data monthly. and we'll compare the percentage of people exhibiting long-term shedding.
And when we look at more recent data through July, we still see, while there's a little bit of a jump in terms of what week patients are in, we still have 67% of patients with their first and their last test in the span of those first 3 weeks. And then that last third just stretches out a little bit longer. So we're still seeing that some of those same patients that are kind of getting multiple tests, it's just stretching out that thing. But the overall distribution, the vast majority of the patients still have their first and last test, and within the span of those first 3 weeks, indicating that we think, that's where the majority of patients line up, and it's just that last kind of third of patients that we really need to monitor and see whether or not this pattern continues as it is, or if something shifts in the future, which might indicate really this reinfection question that we're looking at. With that, I will pass it back to FX.
Great. Well, thank you. And it is those recurring PCR positive patients that are a challenge in the clinic because clinicians need to make a decision about whether this presence of continued PCR positivity represents a risk to others. And so we're going to bring that up in the Q&A in some detail with the combined knowledge on the panel here. Let's go ahead and hear now the story that AJ has to tell us from his amazing analytics team at Entrix. And AJ, I'll just ask you again, I know you were actually on the call last week when we discussed vaccines because you're involved with so much of this emerging science, but please remind the audience what your company does and who your collaborators are.
Sure. Thank you very much, FX, and I'm really happy to be here. And like FX mentioned last week, I had the opportunity to participate on this discussion on vaccines. And this week, I'm happy to be discussing aspects around PCR testing and potential for reinfection. And just to give you a real quick introduction about Entrix. So Entrix is a biomedical data science company, and we're based out of Cambridge, Kendall Square. And we essentially look into different kinds of data modalities, right from biomedical literature, molecular analytics, and real-world evidence, and at a high level, derive insights from both structured data sets and unstructured data sets, and triangulate findings from across these to help address problems of biomedical relevance. And in the context of COVID-19, using the machine learning and AI based software platform that we've developed, Entrix, we've been looking into various aspects of research problems around COVID-19. On the literature synthesis based end, we've looked at coronavirus receptors and their expression, augment that based on NLP-derived associations. And in the context of molecular analytics, we've looked at the spike protein and how it is mimicking parts of the cell surface receptor. And based on real-world evidence, we have a number of studies that we've had the opportunity to work with in the context of a partnership with Mayo Clinic. And in the context of real-world evidence itself, something that is pertinent to this conversation is a study that we've undertaken, and this is looking at long-term SARS-CoV-2 RNA shedding and its temporal association with IgG seropositivity. And it was great to hear the previous introduction from the panelists, and hopefully this will give some additional insights based on that. And this particular study was done in collaboration with Mayo Clinic, Mayo Clinic platform, and Mayo Clinic labs. And pleased to be sharing the panel with one of our co-authors, Bill Morice here, who was pivotal in making sure that we have access to the right data. With that background, the idea of this particular study is to look at the distribution of the PCR tests across individuals and the duration between the time of the first test and last test. As a preface, at the time of undertaking this analysis initially, we had around 75,000 patients. And on the x-axis here, you see the number of PCR tests per individual. And on the y-axis, you have the number of individuals. And evidently, most individuals just took one test. And now if we, with some augmented data that we had on a second run, we had the opportunity to refresh this analysis further. And at this point, over 5,500 PCR positive test patients are available. And here you have the x-axis, the number of PCR tests per patient, and the y-axis is the number of tests for only in the PCR positive patients. And again, a majority of them were administered only one test, but a significant portion of them were also administered two tests. So the availability of this data of patients having two tests gave us the opportunity to look into the duration of the tests itself between the first and subsequent PCR tests. For some reason, my screen is freezing. Apologies for that. Yes. So we took one patient at a time. We had 851 patients that had more than one PCR positive test. And we looked at the duration between the first PCR positive test and the last PCR positive test. And we termed this as the lower bound of viral RNA shedding because this is real world data, and there may be days subsequent to this test that they are testing positive. This is not resolution on a day-by-day basis. So now taking this information into account, if you look at the distribution of the patients, on the x-axis, you have the lower bound of viral RNA shedding, which again is the duration between the first PCR positive test and the last PCR positive test. And on the y-axis, you have the number of patients that tested positive. And in blue, you have the total COVID positive patients, and in red, you have the hospitalized patients. So looking at the distribution itself, the mean duration is around 17.3 days for the lower bound of viral RNA shedding with a standard deviation of 9.8 days. But the interesting part here is that a majority of these patients are actually not hospitalized past, if you look at the time window past day 21 and day 28, majority of the patients are actually not hospitalized. And now, does that mean these patients that are shedding viral RNA are infectious? So that's a separate question to ask. But one can look at other aspects of testing data available, for example, seropositivity, and juxtapose that to get a sense of whether patients tend to shed viral RNA even after converting into a seropositive status. And we had data for about 90 patients where we have this information, which is the time between the first PCR positive test and the last PCR positive test, and the time between the first PCR positive test and the seropositive test. So in purple, you have the seropositive status duration, the upper bound, and in blue, you have the lower bound of the PCR positive status itself. And it is interesting to see that about 14 of these patients, so the x-axis, each pair of bars is a patient, and the y-axis is, of course, time. And when you have the blue bar being higher than the purple bar, what it means is that the patient is continuing to test positive even after converting to the seropositive status. So about 14 to 15 patients from this data set were actually continuing to shed viral RNA even after converting to seropositive status, and these were not hospitalized. And there's one interesting case where you have, like, even after day 80, they were testing positive in the PCR test. So to summarize, we looked at the duration of the duration between the first PCR positive test and the last PCR positive test, first PCR positive test and the IgG test, and the average duration of the PCR positive viral shedding is 17.3 days. And a significant portion of these patients were testing positive even after 4 weeks, and some rare cases of testing even after 80 days. And the majority of these so-called long-term shedders of viral RNA, that is beyond, say, week 3 or week 4, are actually outside the hospital. And the patients can continue to shed viral RNA even after turning seropositive. And like I briefly alluded to, just because someone is testing positive, does that mean they're indeed infectious? So that's a question that needs to be addressed using other methods and other measurements, including looking at viral loads. And I'd love to hear thoughts from the other panelists as well on this. Thank you, and back to you, FX.
Gee, that's fantastic. Thank you, AJ, and thank you for publishing that last month. I know it's, we'll actually put a copy of that paper and the link, a link to the paper in the chat in just a few minutes, but that's really great work, and I'm sure will be subject of additional analysis as your data set continues to grow and become increasingly informative. So I'm going to ask all the panelists now to put their videos on, and we'll start the question portion of our webinar today. And Bill, I think I'd like to start with you. It's for all the panelists, but I'm going to pick on you to ask this. I want to ask this question about: is reinfection possible? And what do we know right now, based on these presentations? It isn't obvious to me whether a person who's had active COVID infection and then recovers potentially could acquire reinfection, and how would we know whether they're actually reinfected or having persistent infection?
Well, it's a great question. Of course, that's kind of the whole purpose of today's session. I think the data that AJ showed really calls into question some of those early reports of, quote, reinfection, because the only way to really answer that question is to actually get documented negative tests, and probably multiple. Again, there's a question in the chat. In terms of going back to the first slide I showed, the challenge with serologic testing is a positive predictive value, meaning some patients that have antibodies are false positive. And so the challenge with the molecular test, by and large, is not on its specificity, but its sensitivity, so the negative predictive value. So the chance that someone that has a negative test truly is negative for the virus. So you have to confirm that they're negative, and then show that they have a new positive, and also would want to see that associated with symptoms. I have heard anecdotally of a few of those actually happening. Again, it's still, though, with these persistent shedders, or persistent positives at least, I don't know that we really know yet, to be completely honest. I think it's still an open question as to, there was an early study out of South Korea that suggested that there was reinfection, but a lot of those I think were probably just persistent positives.
That's really helpful. Currently, the CDC has their recommendations on their website. They were updated just in the last month about returning to work. And I just want to think out loud about, from what you know, Bill, would you incorporate serological testing into that decision, say for a healthcare worker? I think currently the CDC recommends this. Typically their 10-day rule, so 10 days after symptoms have resolved, that it's reasonable to consider having the person return to work, and that if a person had more severe illness, say had required hospitalization, that perhaps 20 days of symptom-free would make sense. But right now, there's no specific recommendation to use antibody testing to make a decision about return to work, and even the use of repeated PCR testing is not something that's required in most settings. So just help us out here. Obviously, the science is emerging, and there may become a role for more lab-based decision-making. But what's going on at the Mayo currently?
So actually, we don't have any testing that's required for people to return to work. It's entirely symptoms-based, as well as other medical conditions. So again, it's pretty much by the CDC guidelines. So if someone's been minimally symptomatic or mildly symptomatic, a 10-day asymptomatic interval, and then if they have an underlying medical condition or immunosuppressed, such that they might have more prolonged active viral infection, then we go to 20 days. I think the absence of testing again speaks to the challenges we have in the testing itself. So, the data AJ shows suggests that we need a test that actually shows who has active viral infection, because there was an early paper in Nature that showed that some people that have PCR positivities, they still don't have active viral cultures, which is not really a practical clinical test. And then on the immune side, we don't do serologic testing because we know some patients, when even the mildly symptomatic who need the least isolation, probably never seroconvert. So we still don't have a great single test to show that someone has generated a competent immune response, right? So what you really need is a test that A, says you do not have an active viral infection that's capable of transmitting, or B, that you have an effective immune response that keeps you safe. None of the tests we have right now definitively answer either of those questions. So we're left with a symptom-based, observational-based system.
Great. And that's pretty much what's being used all around the world, from my understanding. And as the science emerges, we'll start to learn more, whether there's any subcategories of patients or healthcare workers in particular, where more testing would make sense. Ryan, I see Andrea Noel has now joined us from Epic. Thanks, Andrea. I wondered if you could talk a little bit about perhaps Andrea and Ryan and AJ and Melissa about the emergence of data sources for good science, and how do we use information that's used in our electronic health records for the secondary use to understand, the nature of an emerging illness like this, and then secondarily, how do we communicate that even among clinicians, but then also externally to the world?
Andrew, do you want to take a stab at that?
Sure. So there's, I think some of the questions as to, can you determine infectivity? That's probably not something that's going to be easily captured via your electronic medical record, but knowing that a patient has had more than one test or has had positive and then become negative, or persistent viral shedding potentially indicated by multiple positive tests in a prolonged fashion, that's the kind of thing that the electronic medical records will be pretty good at. So being able to harness that through a number of reporting tools, either from an institution perspective or through tools that can tie together multiple institutions, can really let you get a look at data on a larger scale. And in terms of how that can be communicated out to the greater public, I think one of the things we've seen with the pandemic is that there's a lot of different opportunities now for ways to get data to the public. And I don't think there's one right way, but I think some of our leading public health organizations have done a number of things in terms of trying to disseminate data, accurate data out to the public. But there's also a number of potentially less methods of getting data out. Less methods than I saying. We see that Institute Journal, newspapers, online journals, other institutions like that, they pick up the stories and they do take a lot of that data dissemination that may not be, that kind of expands the capabilities of opportunities for getting data out.
Yeah, I think some thoughts there. Really, study design. I mean, we've been spending many sessions. We meet twice a week for an hour, and we go through all the different studies that we're looking at. And it's simple questions can become quite complex when you're working with this data, when you actually try to move it to execution. So things that seem trivial might have a lot of data elements, and we have to kind of confirm between the different partners on what works. I think in terms of communication, in a pandemic, when you got to move fast, I think preprints are great. But then really you should always aim for getting your science out in peer review, and we're certainly pushing for that. I think it's important to call out limitations. If you'll hear me talk a lot about good, better, best. In terms of the good, you want to get it out there, but know that call out your limitations and then say, in the future work, we're going to address these limitations with X, Y, Z, and then execute on that. Just a few thoughts there.
That's good. AJ, I'm interested in some of the new methods that your organization's using for analysis. In this issue about clinical disease, one of the issues we keep hearing is symptoms. And so the data sets that we happen to see now only were the lab test values, but there's a lot of story regarding, was that patient symptomatic? And when we're looking at tens of thousands of cases with these kind of studies, how can we possibly find the information about whether that one patient was symptomatic, coinciding with that lab test value? A traditional, maybe you could tell us, how would that be done in a traditional study, and then what are the opportunities going forward with using real-world information at scale?
Well, that's a fantastic question, FX, and it's also one that is a real challenge of sorts, but also an opportunity that comes with a challenge. Like you mentioned, tens of thousands of tests being done, and it's one thing to have the test outcome, and it's another thing to be able to map the symptoms that go with these test outcomes, right? And when you're looking at EHRs, and of course you have structured fields and unstructured fields in there. In terms of getting information from structured fields, one could basically extract that information in a reasonably less challenging way. But the real challenge is in extracting information from the unstructured data, and that also has aspects of identifying the sentiments around the symptoms. For example, if someone writes, if a physician writes, this patient does not have cough. So it's not a simple search of whether the word cough is mentioned in the EHR clinical notes. And interestingly, Entrix has done work on this very front, and even in the context of COVID-19 itself. So we have like an augmented curation workflow that allows clinicians to tag sentences from the EHR clinical notes, and from a few thousand sentences, the machine learning model learns the sentiments around these symptoms, and then you can go ahead and apply that to tens of thousands or hundreds of thousands of clinical notes, and essentially capture the presence of those symptoms and the sentiments around those symptoms. So on one hand, there's a technological opportunity that we've looked into and applied this to COVID-19 and identified temporal aspects of what the symptomatic signatures are around the testing date itself. So we've had that around day 0, what were the symptoms prior to, in the week prior to the test itself, how is that enriched in the patients that tested positive versus the patients that did not test positive, right? And we found that there's combinations of symptoms such as, cough with loss of smell and taste that are enriched in the COVID-19 positive patients compared to the cohort of COVID-19 negative patients. This is also particularly important that one has like a machine learning or a computational approach to push this, especially in a setting where the frontline workers are resource constrained and cannot devote a lot of time actually looking at individual clinical notes and identifying whether or not someone had some symptoms. So there's a huge operational challenge there that can be overcome with the help of technology.
That's great. Actually, picking up on that idea of the challenges in the technology, I was interested in Andrea and Melissa's and Ryan's thoughts about that collaboration, and how do we create relationships to get good science with electronic health record data. It's not a simple task, and I'm just wondering, Andrea, how can we, how do you attract organizations that want to contribute information, and how do you do that in a way that's privacy protecting?
Yeah, that's a great question. So as Ryan had mentioned earlier, there is a group of folks that meet together on a couple times a week and have been meeting throughout the pandemic. And this includes some the major EHR vendors. So the collective total of folks in the US who have a chart that is encompassed by the folks that are in these meetings, I think that the folks that are on these meetings would cover the charts of a good chunk of the US population. And as we come together and meet and we say, how can we all contribute to the greater good? We are looking at how we can define data elements within our own systems for hospitals who use our software. How can we help everyone who uses our software identify those common elements, the things that we really need to know are consistent across anyone recording a medical chart. So for example, let's just go with diagnosis or the test for the RT-PCR tests for COVID. Do we all know that every hospital is recording this in the same way, in the same place, that we know how to access that in those each individual system's records? And knowing that we have consistency on how we can find the data is such a huge thing in a pandemic. You would think that something as simple as, you know, where does a hospital system store their results for PCR? You would think that that would be so easy. But if we think about each hospital system has carefully built an EHR, it's like saying without knowing, would every hospital system have this data stored in the exact same place? And the answer is no. And so being able to bring all the minds to the table to really know where everything is a big part of being able to move forward with examining bigger patterns, like, are we seeing, you know, evidence that we've got reinfection going on? So once you can identify one infection, you can identify multiple infections, and then as you group together with other colleagues, other data vendors in the field, and establishing those good relationships has been something that's been very valuable over the course of the pandemic. So seeing that you've got folks that in some respects would be considered competitors all working together for the greater good to try to find this common language has just been very valuable. And I think I kind of rambled my way away from the question, but basically, we've got a lot of groups, both in the hospital medical record field, but also our technology partners all around the country, all working together to try to have a common language so that we all know that we're looking for the same thing, the right thing. And then once we have that common language established via these collegial relationships, we're going to really be able to identify those patterns that are going to key us into being able to make a big difference.
Well, thank you.
I think too, once you get that data from all the different sources, you have to look at kind of what's the same and what's different, even within each of those data systems, you know, those stacks of data and those hospital systems. When we looked at the data that we were collecting from Epic, you know, we saw, yeah, we had 16,000 patients that had multiple positive tests, but those were only about 10% of all of the patients who tested positive. You know, we had another 8 to 10% of patients who tested negative after their first positive test, but the other 80% of patients never got tested after that initial positive. So then you have to look at, is there something special about why this 20% of patients was tested again? And then is that the same across different organizations? You know, Bill mentioned that they're not doing repeat testing for their staff members, but are there other organizations that are doing that? Are there other organizations that are requiring repeated testing before patients can be discharged to return home, to return to a long-term care facility? And then you start to get into, well, this is what my data says, but is there something special about this subset of patients? Because, you know, only a small chunk of the patients have this additional field that we're looking at, where the vast majority of patients aren't. So even once you tackle the first hurdle that Andrea mentioned, is you have this additional set of hurdles with the data that you have to try and work your way through as well.
Yeah, that's a great point, Melissa. I think that really understand, first, the two things I would say is, number one, if the data is really key, then we all have to be cognizant that on the one side, the shortage of availability of molecular tests means that we have really under-interrogated and have incentivized to not use the testing as much as we might want to. As a, for instance, you might be, I would definitely biased away maybe from testing minimally symptomatic, you know, in a lot of instances, and that those might be the most important to understand for like reinfection. And then on the flip side, I think serologic testing actually has been underutilized because people don't really understand how to use it in clinical decision making. They see it more as an epidemiologic tool. So we have big gaps that might be driven by biases based on the shortage of availability or lack of understanding and use of the testing, which may or may not be kind of scrubbed out, if you will, by large data sets.
Bill, that's really helpful. And there's a good number of questions in the chat about the issue about viral shedding. And how is that defined? And what do we know about this concept of a virus that is infective versus, say, a virus particle that might be identified in PCR? Is there a way of differentiating those? And I know you've talked about the idea of quantitative PCR testing from a nasal swab or other sources. Can you tell us a little bit about what's emerging there, what might be available in the future?
Well, the traditional clinical microbiology way to document viral shedding is actually to use some kind of culture system, you know, a shell vial culture or something to show that there's actual active viral infection from the specimen obtained from an individual. Those are very difficult to do and certainly not scalable and not widely available. So the press now is, can we identify a surrogate for that, right? And I think the two things that people are holding out are either quantitative PCR, which, because really trying to distinguish whether you have nucleic acid, which is basically remnants, if you will, of the battlefield casualties that are left behind or whatever, versus actual intact virions. And the other then is antigen testing. And we're actually doing that. You know, one of the innovations we've been working on is a high sensitivity antigen test. And we're actually now want to do some parallel studies with antigen, molecular, and some viral culture to see if we can find a correlate, because that's really what we need, is something that's actually scalable to put into a big data sets that can actually answer that question.
Would that imply that the viral capsid, the protein capsid where you're getting that from is what would correlate with infectivity? Yeah,
that's the thought. I mean, the thought is that the antigen tends to be less sensitive because you really need intact virion. If it's not intact virion, then it tends to get proteolyzed quite quickly. So that's the theory, but no one's ever actually, again, this is all new. No one's been that worried about proving infectivity to actually develop a scalable clinical test. And that's, I mean, the pace of innovation is pretty astounding. Someone asked, I would say probably by the end of the year, we'll know a lot more about these things than we do right now. I mean, I would say within, it's not 1 year from now, it's probably in the order of months.
Great, good. And I was wondering if AJ could talk a little bit about how do we use the combination of randomized controlled trials with these real world data sets, given the limitations of each. How do we use both streams of information to try to learn as fast as we can, but with a level of certainty that we can make good clinical policy and good public policy?
That's a really good question, FX. So when you're talking about randomized clinical trials, I presume you're talking about vaccines, right? Or is it drugs?
Vaccines or drugs in this case, or even testing.
Yeah. So with the testing itself, I think it again ties back to when you're trying to get to the efficacy part of these drugs or vaccines. Ultimately, we're trying to get a sense of whether they're getting infected again. And in terms of the PCR positive test that you would get, it would again tie back to whether it's the remnants of the virus itself, or whether you have replication competent viral particles in these patients. Just to paraphrase what was said again, it's going to be a huge logistical challenge, right? To make sure that in addition to the symptom tracking that you're doing and the kind of plaque reduction assays that are used in these in the vaccine related development itself, where you're getting essentially getting samples from the patient and getting to see if the antibodies are neutralizing the particles, is a combination of these PCR tests and the antibody-based test would be ideal, but huge practical challenges remain.
Gotcha. Good. I want to bring people's attention. A few weeks ago, we did have a great presentation from Intermountain Healthcare related to their testing strategy and the fact that some of the availability of different tests is constrained from week to week based on availability of supplies. And I put in the chat, they've shared their decision-making tool regarding that available to other health systems. But Bill, maybe you could tell us a little bit about the constraints on supply. And even with good testing, if it's not available, it's going to be difficult for a health system. What are your thoughts about supply? What are you thinking the field will look like in the next few months?
Right now, I think when we saw the surge in cases across the southern U.S., it became clear, going back to the phases that John spoke to at the beginning. So, starting in early March, it was basically all the major manufacturers of diagnostics focused their supply chains on producing COVID testing. And so we went from essentially zero to where we're doing 700,000 tests a day here in the U.S., which is pretty staggering, right? But the flip side of that is that really required a pivot of all the supply chain infrastructure. And so if you supersede that, there is no backup. And so now the major manufacturers are expanding their production capabilities. So probably by the end of the year, we'll have a little bit more stability in the in-lab supply chain. And I think that's where we'll see a lot of innovation around the more point of care or point of contact diagnostics, whether it's antigen or molecular based.
Great. Well, that brings us to our conclusion. And thank you all for sharing your knowledge, your incredible hard work, and the results that are really starting to emerge now. I'm just impressed with how many great minds are being put to the challenge every day. And thank you all for being part of the coalition and for sharing your work with us. We look forward to having many of you back in coming weeks and months to continue to tell this story. Next week, we'll be talking about telehealth. We have some stories from the front lines that I think will be really compelling for many of you. The week after that, we'll be discussing social inequities as it relates to COVID. Then starting in the fall, we'll continue the series probably at a monthly basis. And we look forward to all of your good input about topics and speakers that you'd like to see. So thanks to all our panelists and thanks to everyone who logged on today. And we wish you continued safety in your work and good health. Thank you.
Thanks, everybody.
Thank you. Thanks, everyone.
Thank you.